Now we're going to get into the different mechanisms of cell injury. So, the cellular response to injury depends on the nature of the injury, how long it lasts, and how severe it is. And the consequences of this injury are going to depend on the type, the state, and the adaptability of the specific cell. So, it is going to vary greatly from cell to cell, but we are going to go over the biochemical mechanisms of cell injury. So, there's a lot of different biochemical mechanisms of cell injury, and we're going to go over each individually. So here they are. We're going to start with depletion of ATP. So here's the effects of ATP depletion, and we're going to go through all of these individually too. So this is just kind of giving you an idea of what we're about to talk about. So the first thing we're going to talk about is the decreased activity of plasma membrane sodium pump. So when you decrease the activity of that plasma membrane pump, what happens is, because this is energy dependent, it's not being activated as much if you don't have the correct amount of ATP, right? And so what happens is you're then going to increase the amount of sodium that's coming into the cell. And as we know, water is going to follow the sodium, so you have the water coming into the cell as well. And so what happens then is the cell is going to swell and you get dilation of the endoplasmic reticulum. Another effect of ATP depletion is that the cell energy metabolism is altered. So if the supply of oxygen to cells is reduced, so like in ischemia, for example, what happens is oxidative phosphorylation stops, and that causes a decrease in ATP, and then you're going to take over ATP formation with anaerobic glycolysis. So that's when you don't have enough oxygen, and that's going to maintain the cell's energy sources by making ATP by using glucose derived from glycogen. So while this is useful in smaller amounts, it's not as efficient, and it doesn't last as long as aerobic. So you're increasing the amount of this anaerobic glycolysis, and that is not only going to decrease your glycogen because you're using the glycogen to make glucose and then to generate ATP, but it also generates lactic acid. So this is important because the lactic acid is decreasing the intracellular pH. And when you're decreasing that intracellular pH, what happens is you're decreasing the enzyme activity inside the cell as well. And this can be harmful to the cell. Another effect of ATP depletion is the calcium pump is going to fail. So if you have the calcium pump fail, what's going to happen is the calcium is going to come into the cell, and that can cause damaging effects on a lot of different cellular components that we're going to talk about a little bit later. Another effect of ATP depletion is structural disruption of protein synthetic apparatus. So basically what happens is you're losing the ribosomes from the rough endoplasmic reticulum. So they're becoming detached, and that is going to decrease the amount of protein synthesis that's happening, and that is a negative effect on these cells. Another effect is that you can have these proteins become misfolded. This is something called the unfolded protein response, and this can lead to cell injury and or death. So obviously that one's bad too. Another bad negative effect is you can get irreversible damage to the mitochondrial membrane and or those lysosomal membranes. And as we talked about in the previous lecture, this can then lead to cell necrosis. So all of these are very negative effects. Having ATP, having the proper amount of ATP is very important. And so all of these can lead to irreversible injury. So remember, in reversible injury, you'll have the cell swelling, you might have some decreased ATP production, and then if this continues over time, it will become irreversible. And these are the reasons why. So this is a nice summary chart from the book. This is from Robbins. So this is showing you here. Let's do laser pointer. So here, if we have a clot that blocks this blood vessel, that's going to block blood flow to whatever tissues and cells are over here. So those cells, the mitochondria within those cells are going to be impacted. They're not going to be getting as much oxygen as they need, and so they're going to decrease the amount of oxidative phosphorylation. So therefore you're decreasing the amount of ATP, right? And so if you decrease the amount of ATP, these are the things we just talked about. So you're going to decrease that sodium pump, which is going to cause sodium to come in. That's going to cause water to follow that. So the cell's going to swell. You can also have potassium going out, which can be a negative effect. And remember, you can have those calcium ions coming into the cell as well. And we'll talk about why that's really bad later. So all of this is leading to swelling of the endoplasmic reticulum, swelling of the overall cell, and you'll get these little blebs. Now, because you're decreasing the amount of oxidative phosphorylation, you're going to try to make up for that by using anaerobic glycolysis. That is then going to use up your glycogen stores, so you're decreasing the amount of glycogen here. And in the process, that is going to create lactic acid. Lactic acid is then decreasing the pH inside the cell, which can cause clumping of the chromatin, which you can see in the cell. And then lastly, the decreased ATP will cause detachment of those ribosomes, which is going to negatively impact protein synthesis. So all of these are negative effects. The next mechanism of cell injury is mitochondrial damage. So mitochondrial damage, what are some causes of mitochondrial damage? And we've kind of touched on some of these already. So first of all, you can get increased calcium inside the cell. We already talked about that. We just said why. So if you are damaging the mitochondria is where that oxidative phosphorylation is taking place. So you'd have decreased ATP. That can then lead to the pumps not functioning properly. You can get calcium coming into the cell. You can also get formation of reactive oxygen species, which we're going to talk about in a little bit. That's a type of free radical, and you can be deprived of oxygen. So again, because inside the mitochondria, that's where that oxidative phosphorylation is taking place. If you have oxygen deprivation, that's going to be depriving the mitochondria of the oxygen itself. It can't create ATP. It's going to then undergo different types of damage. So what are the effects of this damage? So the mitochondria is the powerhouse of the cell. We just saw that it's really important. So if you are damaging these by any of those three mechanisms we just mentioned, what is going to happen? Well, first, what's going to happen is you create this mitochondrial permeability transition pore. So that's a big term, but basically what that means is there's a little hole in it, or a pore. You can also get abnormal oxidative phosphorylation because, again, the mitochondria is where that's taking place. So if your mitochondria is damaged, then it might impair your oxidative phosphorylation. And because you're damaging it, that can actually start to activate apoptosis. So we'll go over each of these individually. So the first effect is that the permeability transition pore forms. So basically, like I said, that's a little hole, and what that does is it's going to cause loss of that membrane potential. And when you're losing that membrane potential, then you're not going to be able to perform oxidative phosphorylation as you should. So if you can't do oxidative phosphorylation, what does that mean? That means that you're decreasing the amount of ATP that's being made. And if you're decreasing the amount of ATP that's being made, we just went over all those different reasons why that's bad. And so that can then lead to necrosis. The second effect that we mentioned was the abnormal oxidative phosphorylation. So if you have abnormal oxidative phosphorylation, what happens is you can actually make these reactive oxygen species. And we just said that that actually can damage the mitochondria itself. And so it's kind of this cycle where if the mitochondria is damaged, it can cause these free radicals, these reactive oxygen species, and those can then further damage the mitochondria. The third effect that we mentioned was that proteins can activate the apoptosis pathway. So they do this by activating apoptotic inducing enzymes. And these, importantly, are called caspases. And you definitely need to know these. We're going to get into these when we talk about apoptosis, which is our next topic. So for now, just know that they're called caspases. And one of the main proteins that activates apoptosis is cytochrome C. So I would circle the cytochrome C and circle this caspase. You should definitely know both of those before we move on to the next lecture. So here's the summary picture from Robbins, and here you can see that if you have decreased oxygen supply or toxins or radiation or anything that can damage these mitochondria, what's going to happen is you're decreasing the amount of ATP because you're decreasing oxidative phosphorylation, and if you have that abnormal oxidative phosphorylation, you can make those reactive oxygen species. And both of these are going to lead to cellular abnormalities, which can then lead to necrosis. The other thing that can happen is if you have these decreased survival signals or DNA or protein damage, that's going to increase the amount of pro-apoptotic proteins and decrease the amount of anti-apoptotic proteins not the amount of them, but the activity of them, which we're going to talk about again in that next lecture when we get into apoptosis. But basically, if you're increasing the uh, pre, the pro-apoptotic activity and decreasing the anti, what's going to happen is you're going to start to undergo apoptosis. So it's this cascade of events that eventually lead to apoptosis. And as that's happening, you can cause leakage of the mitochondrial proteins into the cell, which will further that process of apoptosis. So all of that is going to lead to apoptosis. So in summary, mitochondrial damage can lead to either apoptosis or necrosis. The next main mechanism of cell injury would be that loss of calcium homeostasis. So we already alluded to this a little bit, but what happens? So the cause of this is basically if that calcium pump is failing, that's going to cause the influx of calcium, and then that can damage the cells. So basically what happens is if you have some sort of toxin or if you have ischemia, so again, that's blockage of the oxygenated blood flow to those cells, you're going to have a decrease of oxygen that can then cause the pumps to dysfunction, and you can have increased calcium in the cell. And the reason that happens is because these are going to be released from those intracellular storage of calcium. So you normally have calcium inside the cell, but it's a very small amount. And if you have an injury like this, that small amount will be released inside the intracellular stores, and it'll be coming because those the plasma membrane pumps are not functioning properly. So you'll have an increased influx coming in from the outside. So the inside calcium is being released and the outside calcium is coming in. And what happens if you have this increased calcium inside the cell? Well, it will start to accumulate inside the mitochondria, which is really bad. And it will also start to activate various enzymes that can then lead to apoptosis. So accumulation in the mitochondria. So why does that happen? Basically, if that mitochondrial permeability transition pore is opened, then that's going to cause a decrease in ATP, right? We already went over that. So if you have this calcium that's coming in to the mitochondria, that's going to be mitochondrial damage that is then going to cause, as we already talked about, the mitochondria to open that pore, and then you're going to decrease the amount of ATP that's being made. So that is definitely a negative effect, as we've already talked about. The second effect was the activation of the enzymes. So there's 4 different enzymes that I want you to know the general functions of. So the first one is your phospholipases, and these are going to damage membranes. So think back to cell biology when we talk about, cell membranes and the components of them. Phospholipids are a very important part of membranes. And so the phospholipases, so anytime you see like an ACE at the end of the word, that's typically going to be some sort of enzyme. And so this is literally just like breaking down those and therefore damaging the membranes. The second one I want you to know are the proteases. So these proteases can then break down the membrane and the cytoskeletal proteins. So proteases are essentially just going to cleave protein. So this can affect the cytoskeletal proteins and proteins within the membrane. There's also endonucleases. So these, think endonucleus, so inside the nucleus. So think of what is inside the nucleus. This is going to cause DNA and chromatin fragmentation, because remember, those are inside the nucleus. And then lastly, we have ATPases. So again, ACE, this is going to be an enzyme, and so this is going to pretty much break down ATP. So this is increasing the amount of and speeding up ATP depletion, which, as we already talked about, is a really bad thing for cells. And then the induction of apoptosis. So this happens when you activate these caspases. And so when you start to induce apoptosis, this is going to increase that mitochondrial permeability, which can then cause even further damage. So you see how all of these different biochemical pathways kind of work together. So you're never really going to see just one of these. You're going to see usually a combination of all of these different pathways. So here's a summary slide for that one. So you can see that the extracellular calcium is coming in because of, let's say, damage to the pumps on the plasma membrane. So that is coming in. You also have calcium being released from the intracellular stores. So all of that is leading to increased cytosolic calcium levels. Now, because of that, you're going to get calcium kind of building up in the mitochondria, which will then lead to mitochondrial permeability, which can then lead to decreased numbers of ATP. You will also, when you have increased levels of calcium in the cytosol, in the cytoplasm, what's going to happen is you're going to activate a bunch of different enzymes. So those are the 4 enzymes I just went over, and you should know generally what each one does. So importantly, these both are going to affect the membrane and the cytoskeleton, whereas the endonucleases are going to cause damage to the nucleus. So all of these things, if you're damaging the membrane and if you're damaging the nucleus and you're decreasing the amount of ATP, all of these over time can definitely lead to cell death. The next mechanism of cell injury is oxidative stress. So, what is oxidative stress? What actually causes oxidative stress is basically when you have the accumulation of free radicals. So, you're going to have an increased level of what we call reactive oxygen species. So, this is one type of these free radicals. And so, reactive oxygen species are typically produced normally during mitochondrial respiration and energy generation. So that oxidative phosphorylation inside the mitochondria, but normally these are degraded and removed. But if you have this happening abnormally, you can get an increased amount of these reactive oxygen species, which are a type of free radical, and they can be very, very damaging to the cell. So you'll have an increased production or a decreased amount of scavenging of these free radicals, or a combination of both. So like I said, these are normally made with regular oxidative phosphorylation, but they are scavenged and cleaned up. But if that is somehow impaired, that will then lead to a greater amount of these that can then damage the cell. So these free radicals, they're basically highly reactive because they have a single unpaired electron. And so that single electron can come in, and these free radicals can then attack and modify any molecule surrounding, so proteins, carbs, anything. So those ROS, I would write that down. That's the reactive oxygen species. And like I said, those are the ones that specifically come during mitochondrial respiration. So how do these happen, or how do these accumulate? Well, there's so many different ways. These are just a couple of the different ways. So like I said, normal metabolic pathways like mitochondrial respiration, that's going to cause the reactive oxygen species. You can also have absorption of radiant energy. So that's things like UV light, X-rays. You can have inflammation. So activated leukocytes, especially neutrophils and macrophages. During inflammation, those are going to release these radicals. Then you can have metabolism of exogenous chemicals or drugs. So any chemical or drug that's being metabolized can then cause accumulation of free radicals. Transition metals, so things like iron and copper, and then nitric oxide. So nitric oxide is a normal thing in the body, but if you have too much of it, or if it's in a specific area that it shouldn't be, like that, a lot of this can cause oxidative stress. And so how do we remove these free radicals? Because again, a lot of this is normal activity, normal physiologic activity. So the action of nitric oxide, that's usually fairly normal, but it can cause accumulation of these free radicals. But if you're functioning normally, these are going to be removed. So they can be removed from antioxidants, or we talked about iron and copper. Those should be transported within transport proteins, and so that would help with the accumulation of the free radicals. And then there's also a couple enzymes that can help with decreasing the amount of free radicals in the body. So catalase, this is found in peroxisomes, and this is going to decompose hydrogen peroxide. You also have things like superoxide dismutases and glutathione peroxidase. Now, I don't really care if you know the exact names of those for now, but just understand that there are normal physiologic ways to decrease the amount of free radicals. So if you're not properly decreasing the amount of these free radicals and you have an accumulation of them, that's going to cause that oxidative stress. But what does that actually do? Well, it can cause lipid peroxidation within the membranes. It can cause oxidative modification of proteins, and it can actually create lesions or problem in the DNA. So all of those are very negative. Those can cause cell death. So here you can see the picture from Robbins that kind of outlines this for you if you prefer pictures to words. But you have whatever toxins or radiation coming in, that's going to cause the production of these reactive oxygen species that can then normally be removed by those enzymes I mentioned, so like catalase, peroxidase, glutathione peroxidase, all of those. But if that doesn't happen, if they're not being decreased or removed, you can get those negative effects here. So DNA damage, that can cause mutations. Lipid peroxidation then leads to membrane damage. So all of these can then cause cell death. The next type of mechanism of cell injury is defects in membrane permeability. So we've already touched on this because, again, these are all kind of intertwined. But if you have, or I guess what can cause defects in membrane permeability? So we just talked about reactive oxygen species. If you have any cytoskeletal abnormalities, so we already talked about that a little bit. Phospholipids, remember those are an integral part of any membrane. So if you have a decreased synthesis or an increased breakdown of phospholipids, that can then affect the permeability of the membrane. So what happens if your membrane permeability is altered? Well, the first thing, your mitochondrial membrane. So the mitochondrial membrane is affected because remember that pore is going to open in it. That's going to then cause decreased ATP production, and the proteins that are released can activate apoptosis. The plasma membrane is going to be affected. So if you have a defect in the permeability of the plasma membrane, what can happen is you lose that osmotic gradient. That can mess with the membrane pumps. That can then cause an influx of sodium, which can cause swelling and leaking of the cell that can then lead to cell death that way. You can also have the calcium pump be damaged or not functioning properly, and that can lead to cell death. And then remember, you also have the lysosomal membrane, so if that is affected, that would cause leakage of the lysosomal enzymes, which would then lead to digestion of proteins, RNA, DNA, and glycogen. And so all of that can also lead to necrosis. So here, this is kind of the diagram explaining that. Again, if you prefer the diagram format, but all of this can happen from the membrane damage. And then lastly, we have damage to DNA and the proteins. So we do have a normal mechanism to repair damaged DNA, but it only works to a certain point. So if the damage is too severe and the body can't fix it, that cell is going to undergo necrosis. So here's kind of the summary of all of the different pathways. And remember, these are all kind of integrated with each other. So if you have damage to the mitochondria, that's going to cause decrease of the ATP. That was the first thing we talked about. That has a lot of negative effects. And if you have abnormal oxidative phosphorylation because of mitochondrial damage, that can cause increase in that reactive oxygen species, which would cause oxidative stress. That can then cause damage to lipids, proteins, DNA. If you have some sort of membrane damage that can cause calcium to come in, that can then cause mitochondrial permeability, which can then lead to these things. That can also activate cellular enzymes that can lead to apoptosis. If the membrane is damaged, it kind of depends on what membrane it is, but if it's the plasma membrane, you would have effects on the pump, you would cause leaking, things like that. And if it's the lysosomes, the lysosomal enzymes are going to be released inside the cell, and those are going to eat up all of the cell components that will then cause necrosis. And then if you are affecting the actual nucleus, you can have protein misfolding and DNA damage. So like I said, that can be fixed up to a point, but if it gets too damaged, the cells will then activate an apoptotic cascade and will be killed. So those are the different mechanisms. It is important to understand the different mechanisms, but keep in mind that they all relate to each other. So they're not like, don't think of them as separate entities, but understand each one individually and then try to kind of work your way through it and put them together.